Hello everyone, good morning all. Welcome to biology class. So we can discuss the remaining part of our topic, that is methods of vegetative propagation, artificial vegetative propagation. Okay, so the plant breeders or the plant growers, they developed man-made techniques to make many plants from a single parent plant. This is a usual method of reproduction occurring among almost all garden plants. Okay, this is what is called artificial method of vegetative propagation. The three main methods of artificial vegetative propagation, they are cutting, layering and grafting. So first of all, cutting. Cutting is the most common method that is applicable in almost all type of plants. Usually we are doing this cutting process in case of garden plants such as rose, jasmine, chrysanthemum, etc. What we are usually doing, if we are attracted with any kind of this ornamental or garden plants in our neighborhood or in any area, we are usually, first of all, we are asking for a small stem cutting and planting the same stem cutting in our home. Okay, so after 1 or 2 weeks, the new plant cutting which will start to produce new roots and leaves. This is the most common method of artificial vegetative propagation called cutting. Okay, next is the layering. In this method, the roots are artificially producing on the stem or branches before they get detached from the parent plant for propagation. There are two methods of layering, mound layering and air layering is there. In mound layering, a branch of the plant that is usually growing towards the stem or parallel to the soil, that is usually pulled towards the soil and cover this part of the branch that is in close contact with the soil, avoiding or leaving the tip of the branch as exposed above the ground. So after some time, we can see that new roots are found to develop from the part of the branch that we buried in the soil, and it is very good to make a small cut over or an injury over the stem of that selected branch, which will induce or which will promote the process of development of root very fastly. Then after 2 or 3 weeks or after 1 month, the selected branch that is cut from the parent plant and planted as a separate one. This is the mound layering. So if the plant doesn't have any such kind of parallel branches, so this close to the soil, at that time we can practice the second one, that is air layering. Select any healthy branch from the aerial part of the stem, okay, or aerial part of the plant. Then make a small injury over that healthy branch. Then tie that area by using soil and also polythene. Then treat the part very carefully. Avoid any kind of this infections and others. Care the part, and after 1 or 2 weeks we can see that the process of rooting will be there. Okay, so after the formation of roots, we can cut the branch and plant as a separate one. This is what is called air layering. These are the two methods of layering. Then the another most popular method of artificial vegetative propagation, that is a grafting. In this process, two plants are joined in such a way that they can grow as a single plant or one plant. It's done between two closely related plants. That means two same species only it is applicable. And this process is called grafting. Okay. So in this process, we have to select two plants. One is a plant from which we are taking the healthy stem cutting. So this stem cutting is usually known as a scion. Whereas other one is a rooted plant, one healthy rooted plant. The rooted plant is called the stalk. So in this method, a small part of the stem is removed from the scion by making a slanting cut. Okay. At the same time, the stalk is also want to cut in the opposite way. That means these two branches easily fix each other. Okay, and place the scion over the stalk and fit together by using or tight this part by using a cloth piece, and also over that we can cover this polythene to avoid the infections and others. So after some days the wound may heals and both plants may start to grow as a single one. Grafting is also applicable in almost all type of plants: mango, rubber, apple, then lime, orange, etc. In all this, grafting is applicable. So what are the advantages of this vegetative propagation? It is the simplest and easiest and cheaper or quicker method of propagation. And in this case, the plants which can bear fruits much more earlier than the other type of plants. And also it is very applicable in those plants they are fails to reproduce by seeds, and many plants are produced from a single plant, and we can produce any desired feature of the plant, any desired character. If we want to produce any positive quality or good quality in our plant, we can use this method, and it is very helpful in the propagation of ornamental plants. Then the last method of asexual reproduction, that is a tissue culture. So this is a method of propagation in which helps to obtain a large number of plantlets. Plantlets means seedlings from small pieces of tissues, not from a stem cutting or not from a seed, only from small pieces of tissues taken from any part of the plant, especially from the shoot tip. It's known as a tissue culture or micro propagation. When we are visiting our Anakayam Seed Research Center, we can see this kind of all artificial vegetative propagation methods, grafting, layering, etc. And also this tissue culture lab is also there. So this tissue culture is applicable only in lab. It is not possible in our own yard or in our own surroundings. So the production of plantlets, that is possible only in a lab. Required medium is essential for this method. In this method, the plant cells or tissues are cultured in a special kind of nutrient media which contain all the necessary hormones and also all the nutrients needed for the proper growth of this formation of cell division, fast cell division, and later the cells differentiated to form the tissues. This is the essence of this tissue culture or micro propagation. So the culturing cells that we are selecting from the parent plant, that is usually known as the callus. Then the callus grows very fast and it is then transferred to another medium for the differentiation of roots and also leaves. After the formation of roots and leaves, again it is transplanted in small nursery buds or small pots, and we can take it outside from the lab and allowed to grow until it gets matured. Okay, after matured or after the matured plants is then transplanted to the field or the area that we wish to plant the new ones. It is very popular among nowadays. It is available the tissue culture banana and also a number of grapes, the dahlia flower, the tissue culture dahlia flower, which is showing the fast growth, orchids, chrysanthemum, etc. All these are produced by this tissue culture methods. So these are the details regarding the asexual reproduction methods in the chapter. Then from the next class onwards, we have studied the sexual reproduction in plants and also animals. Okay.